We are live. Welcome back. It is a cold, cold January evening in New York. Best city on earth. Trust me, I've been to plenty. I've been to Dubai, I've been to Abu Dhabi, I've been to LA, I've been to London, I've been to Shanghai. Nothing quite like New York. All right, today we are going to do some biology rapid review. We've got a room of just a few people. More people will trickle in, which is why we're doing a little bit of review before other people get here and we go over sort of higher yield concepts, more important things that we have to do in our lecture. Please come join us if you can, if you're in the area. So, first thing we're going to talk about is very, very, very simple. What is a zygote? What is a zygote? At what point do we refer to something as a zygote? Immediately after fertilization. So a zygote is a fertilized egg. It is one cell. And it begins dividing. But we start calling it a zygote the moment it is one full cell. What do I mean by a full cell? Well, here is an egg cell, and it's mitochondria, right? Because remember, sperm don't have mitochondria. How do sperm get around? Do you guys remember how sperm travel?
The tail, flagella.
So there's a flagella inside the tail, and we need energy to do that. Where does it derive its energy from if it doesn't have a mitochondria? Seminal fluid is full of fructose. Why fructose?
Because it's energy.
It is energy, but why not glucose? What's special about fructose?
It's broken down easier.
When you're at the gym, sometimes if you get super into gym culture, like I was at some point, people will tell you if you need a quick burst of energy, drink what?
Sugar.
Orange juice, apple juice, drink juice, right? Why is that? Well, it's because if you look at glycolysis, so glucose turns into what? What is this on the right? This is glucose. We threw a phosphate on the 6th position, so that's glucose 6-phosphate, right? And that turns into, what is this? Fructose 6-phosphate. And then this is fructose 1,6-bisphosphate. And this is going to turn into DHAP and GAP. So that's glucose metabolism, right? Glucose G6P, F6P, F2, F16, BP, and whatever. We've seen that 100 times, right? That's glucose metabolism. Interestingly enough, fructose metabolism is different. Which you don't need to know, but I want you to know the basis of why sperm use fructose to move around, why fructose is sometimes used in parts of your body when you need rapid energy. Fructose can be acted upon by an enzyme, fructokinase, to make fructose 1-phosphate. And that fructose 1-phosphate can be broken down into two things by an enzyme, aldolase B. Aldolase B. Because this is aldolase, right? This is aldolase, this reversible process. This is aldolase B, aldolase's cousin, right? And it will turn this into GAP and dihydroxyacetone, right? Or sorry, my bad. DHAP and glyceraldehyde. Because the phosphate's at the top, right? And we know this top half turns into DHAP, this bottom half turns into glyceraldehyde phosphate. But there's no phosphate on the bottom. There's no phosphate on the 6th position. We have to put it on there. So then DHAP will go into glycolysis, and the glyceraldehyde will later be turned into glyceraldehyde phosphate. Why is this faster than that.
That's a four-step process because
Not necessarily. What determines the rate of a reaction? It is the rate limiting step. What's the rate limiting step here in glycolysis? PFK1, correct? This is the rate limiting step of glycolysis. Do you see how fructose metabolism evades that step? It skips the rate limiting step of glycolysis. Metabolism of straight fructose skips the rate limiting step of glycolysis. So it is faster. So when sperm are traveling around through what? Through 7-up, right? Which is the seminal vesicle, and then, and then the epididymis, the vas deferens, the ejaculatory duct, the urethra, the penis, right? 7-up. And then the end is nothing, right? When they're traveling through the 7-up, and even inside of the body of, the female, inside of the vagina, inside of the cervix, inside of the uterus, they're surrounded by fructose. They can get around, right? And that's why. Now, I drew something here. On the left, I drew a sperm cell and an egg cell, right before fertilization. I want you guys to tell me some facts about these two cells, things that you know, just off the top of your head. Which one's bigger? The egg. The egg is way bigger. I believe it's the largest human cell there is. I think you can, if you isolated it, you could see one with the naked eye. Don't quote me on that, though. That might be a lie.
I think that's right.
I think I've heard that before. So it's bigger, right? It also contains, almost everything that goes into the zygote, right? But it contains half the DNA. It contains 23N. And this contains 23N. And when they come together, they make 23 2N. What does 23 2N mean? One pair of every chromosome. Because if you say two sets, then it's one chromosome, another chromosome, that's it. This is one pair of 23 chromosomes. And are they single or double-stranded? At this point, they're still single-stranded. And once you start to do mitosis, the beginning of mitosis, this is going to become 46, right? Instead of 23 2N, it's going to become 46 2N. In reality, 92. What do I mean by that? Let's dumb this down into a very basic level. Let's say we have an organism that only has 2 chromosomes. David, nice to see you again. This one's from mom, and this one's from dad. All of the genetic information of this organism is coded in 2 chromosomes. This is 1 2N. One chromatid. When this thing wants to undergo mitosis, right? This is 2N. If I'm incorrect, please quote me, because this is even confusing to me. I read it a bit ago to teach to you. Yes? Good. All right. So there's that. Now, I want you guys to look at this pedigree for me real quick. Squares mean what and circles mean what? Square is male, circle is female. And this means affected, correct? Please tell me the inheritance pattern. Incorrect. Oh, sorry, I messed up. These people are not affected. I messed up. Now tell me the inheritance pattern. It is not X-linked. If it were X-linked, where would it be more prevalent? In the men, correct? Do you want me to draw you one more generation? Let's say we kill this person. These people had no kids. Doesn't matter, they're not affected anyways. It's a square, pretend it's a square. It's going to bother me. What is the inheritance pattern? This is called mitochondrial inheritance. Mitochondrial inheritance. Now, knowing that it's called mitochondrial inheritance, explain the inheritance pattern. It comes from the mother. Every single time a mother has it, she gives it to all of her children. Mother has it, all of the children. Mother has it, all of the children. Why? Because the mitochondria comes from the egg cell. Every single mitochondria on this planet, at some point, came from one person.
I learned about that in anthropology. It's called mitochondrial Eve.
Yep.
Every single human being shares.
Every single human being shares a mitochondria derived of the same person's mitochondria. Not even derived. Mitochondria replicate independently. So it's still that same person's mitochondria. And if you think about it, how many mitochondria does a person have? Hundreds of millions, maybe billions. Ridiculous. It's crazy to think about. This is called mitochondrial inheritance. Speaking of pedigrees, what are some important rules that you should know about pedigrees? So number one is if a mother gives it to all her children, it's possible that it is mitochondrial. Check other mothers and affected fathers to see if they're not passing it on. Number two, X-linked disorders, specifically X-linked recessive disorders, will affect men predominantly. What's a good example of that? Who is a good example of that? Me. I'm a good example of that because I'm colorblind. And I got colorblindness from my mom, who got the X trait from her dad. Autosomal dominant diseases, how do we find those out? Well, let's take a step back and let's look at a Punnett square, right? So let's say we have two parents. Who can name me an autosomal dominant disease? Do you guys know? You read it out of my book today. Huntington's is an autosomal dominant disease. Let's say that this person has Huntington's and they have sex and have children with someone who doesn't have Huntington's. There is a 50% chance that their kid comes out with Huntington's. And then if their kid with Huntington's goes out and has children with another person without Huntington's, there's a 50% chance of that person having Huntington's. So you'll see through the chain of people who have the disease, 50% of their children have the disease. That's how you identify an autosomal dominant disease. And why is the big letter the disease? Dominant. If it's dominant, the big letter is the disease. Do you know what the gene is that's messed up in Huntington's? It's called the Huntington gene. But ironically, Huntington's disease is spelled like this. But the Huntington gene is spelled like that. This is also a really good example of a trinucleotide repeat disorder. Inside the DNA of the people with Huntington's, there is a specific region where there are repeats of a specific sequence. So it's DNA, 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 and then you get this part, CAG, 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 CAG. And if that repetition becomes too long, you got Huntington's. The funny thing is, I don't know if this is on the MCAT. It might be in a passage or something like that. Diseases like these exhibit what's called anticipation. Does anyone know what anticipation is? It gets worse from generation to generation because those repeats get more and more and more. So if your mom has Huntington's, God forbid, and she has 40 repeats, when she passes it on to you, you're going to have 50. And the length of the repeats corresponds to the severity of disease. So it's very common that in neurology, when you study neurology in medical school, you're going to see a family that runs an autosomal dominant disease. And the grandfather got it at 70, and then the mother developed it at 50, and the kid comes in at 37 and into your office with the same symptoms. And that is a textbook definition of anticipation. Good? Very cool. We also call this, in like our joking manner, we call this CAG disease to remember that it's a trinucleotide expansion. All right. So how did we get here? Where were we coming from? We were coming from the fact that the egg has the mitochondria, right? And it passes it on to the zygote. When does the zygote eventually become, not a zygote? It's debated. I think it's like 8. I think 8 is when you start calling it other things, because then it's like a morula and then a blastula and gastrula and blah, blah, blah. When is it a fetus? I believe so. I think so. These are all like really shady, indeterminate terms. But know that there are certain terms you need to know that I don't need to know, that I need to eventually know for STEP probably, but I don't know right now. Morula, gastrula, blastula, fetus, embryo, blah, blah, blah. Embryo is pretty much like the whole time, right? But sometimes people don't like referring to fetuses as embryos, and I'm not going to touch that discussion. Okay, cool. Why did I bring this up? I wanted to ask you guys, right? Let's say that this sperm and egg, they make a zygote, and then that zygote splits into 2, A and B. These are twins, right? These are twins because they split and then they independently grow. So this one independently grows, and this one independently grows. These are called twins. What type of twins are these? Identical. Also known as, medically, what's the term? Monozygotic. They come from one zygote, monozygotic, which means fraternal twins. The other type of twins are known as what? Dizygotic twins. And you will often see, whenever they're talking about twins, you will often see them referred to as monozygotic and dizygotic twins. Very cool. By the way, since we were talking about the sperm cell and how it moves around, what is the specific cellular protein or cellular component that allows a flagella to move? It's part of the cytoskeleton, for a hint. Microtubules, yeah. Microtubules are the important part of flagella that let them move. Very cool. 1 down. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11 to go.
A good question is usually about experimenting with twins.
Twin experiments, twin studies. Those are normally in the psych section, though.
Yeah, I had it somewhere.
Yeah, normally the psych section has stuff on twin studies. What is this? A blood pressure. What is this? Systolic blood pressure. What is this? Diastolic blood pressure. What do either of them mean? Lub dub, right? So it's the pressure at the highest pressure of the cardiac cycle and then the lowest pressure of the cardiac cycle. Now, is this what we mean by, is it 80 throughout the body? No, because we know that at certain points the blood almost stops. There's almost stasis of blood at certain points of the vasculature. We're talking specifically as part of the heart, because the heart is a muscular chamber. It is pumping, right? Everything like this. Now, what are some things that we know? Well, first of all, since we're talking about blood pressure, we have to get it out of the way. BP equals what? Cardiac output times systemic vascular resistance. Systemic vascular resistance, right? And then this is the one that everyone knows. Cardiac output equals what? Heart rate times stroke volume. Systemic vascular resistance. The natural resistance that your blood vessels have to blood flow. Now, last time, in a video I haven't uploaded yet, I equated these concepts to other equations that my physics-minded people would remember. What were those equations? So BEP equals CO times SVR is a lot like V equals IR. And CO equals HR times SV is a lot like I equals Q over T. Take a second to think about that. Give me 1 second. Okay. Another question, just throwing stuff out there. Where do we spend more of our time? Systolic or diastolic? Diastolic? Systolic? Malcolm? Diastolic. We spend more of our time diastolic, about 2/3 of it. Because if we spent more of our time in systolic, you'd probably feel your heart gasping for breath. Right? Okay. Good. That's basically all you have to know about systole and diastole. When you get to medical school, this becomes much more complicated. Now remember, I wanted to ask you guys one thing about this equation right here. We know that cardiac output is part of blood pressure, correct? And we said that stroke volume is part of cardiac output. Now what's part of stroke volume? Total body water, right? Because the more water that you have inside the body, the more blood you have inside the vessels, which means the more stroke volume you have because you have to push that amount of blood through the body at the same rate, which means your heart fills with more water, right? Remember that thing I told you guys to do way back when, I think this is like chapter 3, 4 of bio. I told you guys to hold your hands together like this and just generate pressure between them, right? And you can't do it. You can't generate pressure in the middle of that hole. Because why? Because there's nothing there to create pressure against. You need something in between your hands to create pressure against. But if I take this water bottle and I put it between my hands, now all of a sudden I can create pressure against this. And if the top wasn't here, this would be actually bursting with water. That's what your heart does. The top is open, or it generates enough pressure to open the top, and the blood goes flowing out the top of the chamber, right? So you see two ways in which the heart can increase pressure. Number one, it can increase pressure just by pumping harder, right? Which is contractility, which is part of the sympathetic nervous system. Sympathetic nervous system innervates the heart, right? And allows for increased contractility. The other thing is to increase the amount of stuff it pushes against. If you increase the amount of blood inside of the heart chamber, you can increase the pressure it generates, right? And how do we do that? You guys know all about the renin, angiotensin, aldosterone system, and ADH, antidiuretic hormone, which serves to conserve water, increasing the blood volume and blood pressure. The RAS is more about blood pressure than it is anything else. And ADH is more about controlling blood what? Osmolarity. Because the things that signal for ADH release are sensitive to blood osmolarity. When blood osmolarity gets too high, ADH is released, aquaporins are sequestered into the collecting duct and distal convoluted tubule of the nephron, and that increases water reabsorption and retention. It's how we regulate our sodium balance. What else is going to happen when your blood osmolarity is too high? You're going to feel thirsty. You're going to get thirsty. Very good. What is this? The bicarbonate buffer system. Very good. And it buffers the blood at a lower pH range because the pKa here is what? 6.2. 6.2. Well, remember, we're still in buffering range because we're within 1.5 of the regular body pH. What is the regular body pH? 7.4. 7.4. So the majority of this is what? Associated or dissociated? The majority is dissociated. Remember, higher pH that way, lower pH that way. Right. There's an enzyme here called, does anyone know what it's called? The enzyme that does this? Carbonic what? Anhydrase. Carbonic anhydrase. Now Sarah, who will be here later, was so kind enough to share with me a question that she saw. And the question was, if you inhibit this enzyme, what's going to happen? That enzyme specifically, inhibit it in red blood cells. Inhibit the carbonic anhydrase in red blood cells. What's going to happen? Kira, how you doing? So, if you inhibit carbonic anhydrase in red blood cells, could you sit there? Here, in the front. Could you just move that over for him? Because he finds it quite difficult to read my handwriting sometimes. If you inhibit carbonic anhydrase in red blood cells, which of the following will happen?
Is it a multiple or just one?
Just one. Well, let's think about it logistically. This one is clearly wrong. Because if you inhibit the enzyme that makes it, you're not going to push it forward. That's not going to happen. Clearly wrong. That's what you want to do with the MCAT. Look at stuff that is blatantly, obviously wrong and immediately get rid of it. There are too many times that students look at questions on the MCAT and say, oh, well, you know, like if this were the case and this was this, maybe that'd be correct. No. If you have to jump through loops of fire to get to an answer, it is incorrect. That's true of everything in life, especially relationships. Do not do leaps and bounds to make things work. Decrease oxygen affinity. Well, we have to think about what decreases and increases oxygen affinity, right? We need to actually actively think about that. So that's not blatantly obvious to us yet. Increase CO2 in tissues. Well, I don't really know, without thinking about it, what RBCs do for CO2 in tissues yet. So let's come back to that as well. And increase HCO3 minus in RBCs. Well, we just said this isn't going to go up. So if that doesn't go up, that's not going to go up. If anything, it's going to go down. We're between two choices. So now, like many times, we have a 50-50 shot at guessing right. So if you're short on time, you just bought yourself a 50-50 chance of getting this right instead of 1 in 4, right? But we're not short for time because I could be here all night. We'll figure it out. Okay, so the decreased oxygen affinity, right? What do we mean by oxygen affinity? You remember this? You remember this? And there were things that shifted to the right. And there were things, Sarah, we're talking about your question. And there were things that shifted to the left. Guys, this is Sarah, if you haven't met her before. We have new people in the front. And that's Kiro, that's David, that's other David, that's Malcolm, and Joseph. And Ihtisam, right there. And there's Mustafa. Habibi. I know, right? It's all going to be gone in like 2 weeks. Some people are jacking your spot, bro. You got to fight them.
I know, I'm here already.
So we have effects that push the hemoglobin-oxygen dissociation curve to the right and push it to the left, correct? We know how pH affects the hemoglobin-oxygen dissociation curve. How does it do it? When the pH goes down, there's more loading or unloading? There's more unloading when the pH goes down. Why does that make sense? How are you going to think about that? How are you going to remember that? I'm saying when the pH goes down, it's going to shift to the right. How do we know that? Because shifts to the right are offloading oxygen, correct? And when is a case where the blood pH inside of a human body goes down? During exercise, lactic acidosis. During exercise, the body pH goes down, and you have to think we want more oxygen offloading at that time. So this decreases oxygen affinity and increases oxygen offloading at the tissues. So an increase in oxygen affinity would be caused by a increase in pH. Correct. So, what's happening inside the red blood cell? We're holding on to this. We're holding on to this. We're not making H+, which means the pH isn't going down, right? So the pH isn't going down, then what? The pH is going up. And that means it's increased oxygen affinity, not decreased, because the pH would have to go down to decrease the oxygen affinity. This is incorrect. So the answer is C. But why? What does an enzyme inside the red blood cell have to do with CO2 in tissues? Well, you need to know how red blood cells work. Feel free to move forward if you like. I write a little small, I know that. There's a seat right here, seat right there. How do red blood cells work? Because obviously they swap the oxygen for the CO2, right? How do red blood cells work? The question isn't how do they work, because anyone could tell you that. Oh, it drops off the oxygen, it picks up the CO2, and it leaves, and then it goes to the lung, yay, right? Like anyone could tell you that. A fifth grader could tell you that. But how does it actually work? Well, the red blood cell picks up CO2 from where? The tissues, from respiration. And the CO2 isn't held as CO2. It uses carbonic anhydrase to turn the CO2 into H2CO3 and turn that into bicarb. And this is the fact. This is what all of this was leading up to. The majority of CO2 carried by red blood cells is carried in the form of bicarb. The majority of CO2 carried by red blood cells is carried in the form of bicarb. So if you inhibit carbonic anhydrase, you will limit the amount of bicarb that can be made. You will limit the amount of CO2 that can be carried, and you limit the pickup of CO2 from the tissues, thus increasing CO2 in the periphery. Yes?
I'm guessing you wouldn't have that graph on the question, or would that be?
You would not have this graph on the question. You would have to be in your head.
I know that using this type of elimination process, you don't see oxygen there, so I would kind of ignore it. But I'm
Right.
need to know
Right.
the whole thing. But
Exactly.
I also don't see it there, so I would automatically.
I think the kicker to think about oxygen and how it plays a role in this is this. The fact that it's happening inside of red blood cells.
How did you discern there was an increase in pH?
Sorry?
How did you discern there was an increase in pH rather than staying at 7.4?
Oh, because if you can't use carbonic anhydrase, the whole reason that CO2 decreases pH is because the CO2 turns into H plus and HCO3 minus, and the HCO3 minus goes away, right? It's the whole reason that that happens. The reason I said that there was an increase, hold on. First of all, not necessarily that there's an increase in pH. I'm saying that to decrease the oxygen affinity, we have to decrease the pH and make more H plus. Since that's not happening, there is no decrease in oxygen affinity, is what I meant, right? I said theoretically if we were to go the opposite direction. That's just a logic thing. That if you want to disprove something, you prove that the opposite is true, right? That's something we did a lot in philosophy. You want to prove a point, prove that the opposite is or could be true. Sorry, you want to disprove a point, prove that the opposite is or could be true. All right? The question was, if you inhibited carbonic anhydrase inside of red blood cells, which of the following would happen? Carbonic anhydrase, fun fact, is also the most catalytically efficient enzyme in the entire body. What is catalytic efficiency? Kcat over Km. Or sorry, yes, that's catalytic efficiency. Kcat over Km. Wonderful. Very good. That's the bicarb buffer. What's the other buffer we have in our body? The phosphate buffer. And since I told you the pKa of the bicarb buffer, we can look at the pKa of phosphate buffer. We start with phosphoric acid, which turns into monobasic phosphate, which turns into dibasic phosphate, which turns into phosphate. And for polyprotic acids that kind of dissociate in this manner, this is the rule, that the pKa will increase by approximately 5 points for every deprotonation, from 2.2 to 7.2 to 12.2. So I ask you, what is the predominant species of phosphate in your body? A, B, C, or D? What is the predominant species of phosphate in your body? It would be not B. C. Why C? So 7.2 is right here. 50-50, right? Why do we know that? Because pH equals pKa plus log A minus over HA. And if A minus and HA are equal, then this comes out to 0, right? And pH equals pKa. So if the pH 7.2 equals 7.2, right, this would be 50-50. But it's not. We're saying that 7.4 equals 7.2 plus log blah, 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 blah. And in order for this to increase that side, this needs to be predominant. We need more of this. And how are we going to think about that? Well, here's 7.2 on a number scale. Where is 7.4? To the right or to the left? To the right. Very good. Lots of new faces today. Guess people are really starting to study for the MCAT now. Either that or you guys are doing your job and you're telling people about this program. Who's here on referral? Cool. I am too. I referred myself. I referred myself to be your teacher. I kind of did. All right. Before I move on, now that there's a significant number of people here, is there anything out of any of the, you guys can go look back in your notes while I prep the next 10, 15 minutes of the lecture. All of you guys who have been here the whole time or have been taking notes, go back into biology, look for something that's confusing, and I'll explain it. If you guys would like me to explain anything at all, I will explain it today. Let's talk about digestion real quick, just for a bit. And one specific part of digestion, the anatomy. Because unfortunately, some of us get fucked by digestive anatomy. No pun intended. So what's the first part of digestion? The mouth, correct? We go from the mouth to the oropharynx, which is just that back part of the mouth right there. And then we go into the esophagus. The first part of the esophagus is pretty stiff, right? Hard. And then you go down and it becomes smoother muscle. Is the esophagus always open, by the way? Is the esophagus always open? No, it has to be forced open. And I learned that when I was doing anatomy. The esophagus is normally just this closed little loop like this. And when you swallow, it opens up and then closes again. It's interesting. What is the thing that shuts the trachea when you swallow? What's it called?
Epiglottis.
The epiglottis. Very good. It moves forward and closes off the trachea when you swallow, and food just slips past the epiglottis.
I also forgot, this made me remember from anatomy that the reason why you can also swallow things upside down is because of the involuntary muscle movements.
Peristalsis. There is involuntary smooth muscle contraction all along the GI tract. That is called peristalsis. You want to give it a shot? Jump out of an airplane upside down and swallow a chug of water. Just bring a parachute. All right, so the esophagus. The esophagus leads into the stomach, the upper part of the stomach. And the stomach has 4 parts. And we're going to write out the 4 parts. So let's just say this is the the gastroesophageal junction. The gastric sphincter, right? Whatever it's called, I forget. I should know this, but I don't. So the first part of the stomach is called what? The fundus. Then we have the body. And then the antrum. And then the pylorus. Do you guys like, I don't know if you guys like it or not, so I won't do it if you don't like it, but do you guys like it when I give you medical knowledge that relates to your lesson? Yeah? There is a disease in children called pyloric stenosis. Pyloric stenosis. And before you move on from the pylorus, you need to pass the pyloric sphincter. And it is a muscular sphincter that controls the output of gastric contents into the, what's the first part of the small intestine? The duodenum. So before things get out into the duodenum, they have to pass through the pyloric sphincter. What is stenosis? What is aortic stenosis, mitral stenosis, pyloric stenosis? It's a closure, right? It's like a stiffening, right? Aortic stenosis is a stiffening of the aortic valve, so blood can't really get out of the aorta. Pyloric stenosis is a stiffening of the pyloric sphincter, so it stops contents of the stomach from getting into the small intestine. These kids are going to projectile vomit out the stuff that they eat because it can't get down. And the stomach fills and fills and fills, and it causes them to gag all their food out. This needs to be surgically managed, and the surgery is called a pylorectomy. You cut away muscle from the pyloric sphincter. So, this is now the small intestine, and this is the stomach. So we have the duodenum, and the duodenum leads into the jejunum, and the jejunum leads into the ileum. The ileum is the absorption point of vitamin B12, specifically. It's pretty late.
That's where you get like the issues with intrinsic factor and B12 deficiency.
Mm-hmm. Very good. Intrinsic factor is required for absorption of vitamin B12. It's a very characteristic anemia that occurs when you are deficient in vitamin B12. It's called megaloblastic anemia. The ileum, before you move into the next part, you have something called the ileo-blank valve. The ileo-cecal valve. Because this next part is called the cecum. And this is now the large intestine, otherwise known as the colon. The colon. Does anyone know what buds off of the corner of the ileocecal valve there's this little outgrowth of tissue at the corner of the cecum. I've seen it. I've held it. The appendix. Anyone know someone who's gotten an appendectomy? A lot of people. Lots of people know someone who got an appendectomy. This is probably the most people we had in class before. That's nice. Look at that. Tell your friends, everyone. Tell your friends to come. If they want high-yield MCAT facts thrown at them like boiling hot water, please don't throw boiling hot water at anyone. This is going to become like the new don't sniff, what was it? Don't sniff acetone. Because we're talking to Orgo about acetone, and I was talking about how much I love the smell of acetone. Don't sniff acetone. The cecum. Then we have the 4 parts of the, well, technically there's 5 parts of the colon because the cecum's one of them, right? And where is this in the body? If you guys could point to my body, where is, or your body, where is in the body? It's right here, the ileocecal junction, right here, right? So the cecum's gonna bend up towards the body because what's weird is that we have the stomach here and the small intestine just loops and loops and loops and loops and then throws you right here. And instead of going straight to the ass, we do this whole roundabout thing. We do a whole fucking turn around the body because we go under, up, over, down, and out. So let's think about that again. And the reason I say this, the reason I orient you, is because the next part of the colon are referred to by their position. So first we have the cecum, which curves us up like a little C. And then we have the part of the colon that goes up. That's called the ascending colon. Then next, we have the part that goes to the left. This is the transverse colon. Next, if we had an ascending, we have a descending colon. And lastly, we have something that loops down and puts us towards the rectum, the sigmoid colon. What does a sigmoid curve look like? Looks like that. That's kind of what the sigmoid colon looks like. Just not as 2D, hopefully. And the sigmoid colon leads us into the rectum, or the rectal vault. So, why? Why? What's the point? Why don't we just go? We're right here. You just got to make a loop backwards and go. Why? Why? Why does the colon exist? Because what's absorbed here and what's absorbed here? Nutrients, sugars, fats, proteins, vitamins, etc. Out of everything I just named, what did I not say? Water. Water reabsorption. Or absorption, not even reabsorption. Water absorption. Water is constantly being pulled out of the gastric and intestinal contents. Pulled and pulled and pulled and pulled. Do you know how much water there is in a fucking mushroom? You guys ever eaten a mushroom before? There's so much water in there. You are pulling it out of all of the food you eat, and all the meat, and all the beans, and all the lentils, and all the everything. Everything that you eat that has any amount of moisture in it, as long as you're not eating fucking chalk, it has water in it, and you're pulling it out from the food you eat. Because if you weren't, you would die. Either you'd die or you'd be pissing out of your goddamn ass. Which happens when you have diarrhea. Because the bacteria in your gut fuck up your absorption capability and shit just goes and you start squirting out of your anus. You start painting the fucking wall brown. So if you have a problem up here, your stool is going to be fatty. You're going to have a problem with fat reabsorption. You'll be able to see it. fatty stools are a really bad sign in medicine. There could be something pretty bad going on. If you have a problem here, you can have watery stools.
Where would Imodium come in?
Imodium?
Yeah.
Is that like, what was Imodium again?
Yeah.
Antidiarrheal, right? I think it's over here. Oh yeah, so it's like a osmotic agent, and it causes whatever. So if you increase or decrease the osmolarity of like a specific substance inside of here, you can increase or decrease the amount of water that goes in or out. Like a laxative. A laxative is going to be like fiber. It just pushes everything along. Since it pushes things along, it decreases the amount of time you spend here. It waters down the stool, and it makes it easier to pass. I actually haven't read up on Imodium in a while. It's been like 7-8 months, so I'll get back to you on that. Stay hydrated, folks. Was that a good overview of digestion? Are we all sufficiently disgusted? Okay, good. Wonderful. Does anyone have anything they want me to explain to them? Or did you guys forget that I asked you that question? Go for it, Kira. We're all waiting. Clock's ticking. Gen Bio. That. Do you see the fucking title, Sarah? God. What's up?
Where's diverticulitis in the stomach?
Diverticulitis. Well, diverticulitis isn't a stomach problem. It's a colon problem. So what's a diverticula? A diverticula is basically like you have the colon right here. Get a little outpouching like that. It's a diverticula. Yes? Oh, brown fat. Do you not remember we were talking about brown fat last time? Thermogenin. I'm surprised it hasn't been erased yet. Babies have a lot of brown fat for thermogenin because they have to keep warm, because they're idiots and don't know how to survive. You actually just talked about something that's on my list to cover today. So let's talk about endocrinology. Let's talk about the center of endocrinology. Who is head honcho of endocrinology? Almost. The hypothalamus. The hypothalamus. This word sends shivers up my spine because I just took my neuroanatomy practical. And the number of times I've had to identify the hypothalamus, if I had a nickel, I would probably have 25 nickels. Exactly. I would have $1.25. It's probably more money than what's in my bank account right now, because I don't get paid to work here. I love making that joke. So, what are hormones released from the hypothalamus? Well, as an MCAT student, it is a bit difficult to think about it from the position of the hypothalamus. So maybe we should start at the pituitary. Right, so the pituitary hormones are flat, PEG, from the anterior pituitary, and OA, from the posterior pituitary. So this is anterior and this is posterior, front and back. And the pituitary, it looks like a little ball sack, right? And then you have the pituitary stalk, which leads up into the hypothalamus, right? This is the anterior and the posterior pituitary. What did I tell you the posterior pituitary was? It's just one giant neuron. It's one huge neuron. Because axons from the hypothalamus will come and invade the posterior pituitary and just send out O and A. What are O and A? Oxytocin and ADH. Oxytocin is good for one thing on the MCAT and one thing only, and it's not fucking. So what's the other thing? I just said not fucking. labor. This is the only example of a positive feedback loop that you need to know. What's the positive feedback loop? When a baby is ready to be delivered, it will start trying to crown. It will push up against the cervix and cause cervical dilation. What is the cervix? Well, these are the fallopian tubes, and this is the uterus. How big is the uterus again? Did I tell you guys when I dissected a uterus? It's about that big. It's tiny. It's really small. And that thing that's this big and about that wide in dimension carries a whole human inside of it. It's ridiculous. Shout out to our women. All 2 of you in the room. Let's go. Here is the end of the uterus, and the junction between the birth canal, or the vagina, and the uterus is the cervix. It is a very small hole that sperm can pass through. It is a muscular junction. I believe it's muscular, right? And what's going to happen is the baby is going to come out of the uterus and push up on the cervix, and that's going to cause cervical dilation. That cervical dilation will signal the hypothalamus and the posterior pituitary to release oxytocin. Oxytocin will hit the smooth muscle of the uterus and stimulate its contraction. And the stimulation of uterine contraction will dilate the cervix, cause more oxytocin to release, more contraction, more dilation, more oxytocin, more contraction, more dilation, more oxytocin, more contraction, until the baby is out and the cervix closes back up completely. Well, after the placenta and everything comes out, right? Cervix bounds back, uterus comes back to regular size, and now the woman is chock full of this cuddle hormone. And the first thing she does is hold this human being that she doesn't know, and boom, immediate bond formation. So this is the evolutionary process by which we have developed bonding between mother and child. Some also stipulate that the drop in oxytocin after labor is a big part of the reason for postpartum blues.
What if the kid's C-section, would the mother still feel that?
Yeah, because there's still a natural sort of progression there.
Does the child also release oxytocin to a certain degree?
I don't know. I have no idea. But there are some pretty cool reflexes inside of newborn babies, inside of babies in general. If you stroke the cheek of a baby, it'll turn to that side because that's like a breastfeeding reflex. So when the nipple touches like the corner of a child's cheek, it'll turn towards it. It's called the rooting reflex. Another one is the grasp reflex. If you guys ever have been near a very, very young child, you put your finger in its hand, it'll close its hand around your finger, and it'll actually have quite strong grip for a fucking baby. Also, if you put a baby underwater, it will instinctively hold its breath. Another one is the Moro reflex. It's like the startle reflex. So if you take a baby and you hold it, don't do this. don't do this. This is done by trained professionals inside of a hospital setting who are ready if the baby falls. You hold a baby like this, and you drop your hand a couple inches, and the baby falls back, and it spreads itself wide like that to protect from a fall. It's cool, right? Okay, so that's the whole thing, oxytocin. What is ADH again? We just talked about ADH before. Responds to high blood osmolarity, right? Waters down the blood, increases blood volume, et cetera, right? These are the bane of every MCAT student's existence, unless you use Anki, in which case you know these by heart. Let's go through them. So first, I'm going to kill myself. Oh, damn it. I keep forgetting all this is on camera. Seek help if you need it. FSH, right? Follicle stimulating hormone. What's all that yapping? No, I'm kidding. I don't give a shit. I literally don't care. Follicle stimulating hormone, right? FSH. Who has follicles, men or women? And when you stimulate the follicles, what do? They release estrogen. Follicles release estrogen. So if it releases estrogen in women, does it release testosterone in men? They're opposite. It doesn't release testosterone in men. What does testosterone in men? Luteinizing hormone. This does testosterone in men. This does estrogen in women. Which means this luteinizing hormone does progesterone in women. And follicle stimulating hormone does what in men? Spermatogenesis. DHT is a testosterone derivative. And it's the reason you and I are going to go bald someday. Unless you book a flight to Turkey, you pay a guy $1,000. Follicle stimulating hormone and luteinizing hormone. I would love to go over the menstrual cycle again. I have a whole 28-minute lecture about it on my YouTube channel. Watch it if you have to. It is part 2 of chapter 2 of general biology. It was uploaded late because I had to re-record it. My audio cut out the first time. So what's the A in flat peg? And give me the full name. No ACTH. None of that. Adrenocorticotropic hormone. Adrenocorticotropic hormone. ACTH. Break down the name. Adrenocorticotropic. Adreno meaning adrenal gland. Cortico meaning cortisol. Tropic meaning it gets someone else to do its bidding. And then, yeah, tropic. And what's doing the bidding? Cortisol. It is a corticotropin, right? So what does ACTH do? When it's I love how I just had the drawing of the ball sack on the whiteboard for like the past 15 minutes. So adrenocorticotropic hormone is released from the pituitary in response to what from the hypothalamus? No, there is a hormone that controls the tropic hormone. CRH. Corticotropin releasing hormone. I think it is. Yeah, it's CRF. That's what I was thinking about. I was like, there's something wrong. Corticotropin releasing factor. My bad. CRF stimulates the pituitary to release ACTH. ACTH circulates through the bloodstream and hits the bean and his friend. Who's the friend? The adrenal gland. Now, the question I ask every single student, and they hate me for it. Which part of the adrenal gland is it hitting? The outside or the inside? And if I called them by their names, the answer would be obvious. The outside. Why? Because the outside's called the cortex, and it makes cortisol and other steroid hormones. So it's going to hit the cortex and release cortisol. If I were to zoom in on this, which you will do in medical school, this is the cortex, and this is the medulla. Similar to the posterior pituitary, the medulla is one ginormous neuron, one huge neuron. And this is the site of what synthesis? Give me the fancy word. Catecholamine. Catecholamine synthesis. And the cortex has 3 layers. The first layer does salt. The second layer does sugar. And the last layer does sex. There is a constant orgy going on in that third layer. Just pound town. No, but it actually controls the production of a very specific androgen called androstenedione, which is important more for women than for men, because men produce most of their androgens in the testes. Women are dependent, a little bit more than men, on the function of the third layer of the adrenal cortex for the production of androstenedione to turn into estrogen. And then catecholamine synthesis down here. What are the catecholamines? Norepinephrine, epinephrine, dopamine. More specifically, norepinephrine and epinephrine. Cool. So cortisol released from the second layer of the adrenal cortex. Cortisol regulates what? Sugar balance, correct? And stress. This morning, when I was awake at 6:45, right? Because I had my exam, I woke up at 6:45, and I walk into the exam hall, and my friend Dan walks in. Great guy, I love him. He's a really, really nice person. Wants to be an ophthalmologist. I hope it works out for him. He comes in, and he's like, I just had so much coffee, man. So much. So awake right now before the exam. And I am sitting there trying not to fall asleep at 8:15 in the morning. It's about, like, orientation's at 8:30 for the exam. And I'm just like, I'm waiting. And he's like, what are you waiting for? I was like, I'm waiting for my cortisol to kick in. Why? Why should you not drink coffee first thing in the morning?
Because you're already tired.
Because when you wake up at hour zero, what wakes you up? Cortisol. If you wake up naturally, cortisol wakes you up. And it slowly starts to climb. And it hits a peak about 2 hours after you're awake. And then it just slowly starts to dip, and it slowly starts to climb again. Which is why you'll realize, like, 7-8 hours after you've been awake, you're kind of tired. Like 2, 3 p.m., 4 p.m., you're kind of tired again. But if you make it past that, if you make it to 7, 8, 9 p.m., oh my God, what time is this lecture? Wow. Oh, man. Did I coordinate this lecture to be part of your natural sleep-wake cycles? No, I didn't. This is just when I was free. But if you make it past that, you're fine. You hit your second wind. The cortisol kicks back in and wakes you up. Right? Yeah, so if the thing is, if you drink caffeine here, yeah, you're going to hit a high. You're going to hit a high, but this isn't going to feel like awake anymore. So if that doesn't feel like awake, the fuck is this going to feel like? You're going to feel like shit, my guy. You're going to feel like garbage.
I don't know if you listen to Huberman. He says not
He's
to
great.
drink caffeine, like wait 2 to 3 hours after you wake up.
Exactly, because your cortisol levels spike 2 hours after you wake up.
So how about morning people? Because they're not normal. People that just wake up and are fully energized.
Morning people?
Yeah.
I mean, like, everyone has a different level of sort of, like, cortisol, blah, blah, blah. And your body adjusts to whatever you do. So if you're a morning person, you've been a morning person, your body's going to adjust to that, right? I'm not a morning person. I'm most certainly not a morning person. I wish I was. But that's also why I want to become a surgeon, because I can just be like, oh, you guys need me to work until 5 in the morning and then go home for the rest of the day? Yeah, sure, why not? I don't give a shit. Okay, adrenocorticotropic hormone. The T. TSH, correct? What is TSH? Is it thyroid stimulating hormone? Or is it thyrotropin? No, it's thyroid stimulating, yeah. What was thyrotropin? That was TRH, thyrotropin releasing hormone from the hypothalamus. So before we get to the hypothalamus, let's figure out this. Flat, and what's the peg? The 3 kind of unknowns, right? Prolactin, endorphins, and growth hormone. I learned this unit in medical school, that endorphins actually act on the same receptors as opium, heroin, codeine. They're literally just natural opioids. It's crazy. It is called the, if you want to do research on it, it is called the mu-opioid receptor. The mu-opioid receptor. By the way, what does mu mean in physics? Micro. So what's a micrometer? 1 times 10 to the what meters? -6. And then what is this? What's this? And then what's this? -1. What goes in between here? Centimeter. And what's this? And then after that, I think it's femtometer. I think that's 15. Cool. Someone had a question? No? Okay. Okay. So, talked about that. Okay, prolactin, milk production, and what? Inhibition of other sort of menstrual and sexual hormones. So we talked last time about a patient that I saw in a neurosurgery clinic, 37-year-old man, very young, healthy, works out a lot, et cetera, came in with erectile dysfunction. 37-year-old guy. Erectile dysfunction and headaches. And he was what? He had enlargement of his breasts. Gynecomastia. Nope. He had a tumor. And that tumor was secreting prolactin, which was killing his sex drive, causing gynecomastia. Yes?
So here the tumor, I know that some tumors aren't cancerous, right?
Mm-hmm. So that's not a cancerous tumor. A prolactinoma is a benign tumor of the pituitary. So that's called it's of a subtype of tumor. It's a subtype of a tumor called a pituitary adenoma. And usually OMA means not malignant. A pituitary adenoma, right? And there's a bunch of different types of pituitary adenomas. You can research them. They're quite interesting. But this one secreted prolactin. Right. How do you inhibit prolactin? We have a whole prolactin inhibiting factor in our body, also known as dopamine. Very good. Dopamine. Endorphins, painkillers, and growth hormone causes linear growth. Okay. A, B, C, and D. Who can tell me what secretes what? What secretes A? What secretes these two? Huh? The full name. Gonadotropin releasing hormone. What secretes B? We talked about it before. Corticotropin releasing factor. What secretes C? This is one a lot of people forget. Thyrotropin releasing hormone.
Can you repeat A again, please?
gonadotropin releasing hormone. Give you a moment to take everything down. Okay. Gonadotropin releasing hormone, corticotropin releasing factor, thyrotropin releasing hormone, and the last one. What secretes growth hormone? This is the easiest one. Growth hormone releasing hormone. Very good. Yeah.
I thought BDF also secretes growth hormone. What is it, like brain-derived or something?
No idea. I wish I knew. But I do know that the secretion of growth hormone and growth factors in general is a very complicated study. If I were only an inch taller, I'd be 6 feet tall.
Should have taken growth hormones.
Exactly, right? Should just start fucking shooting up growth hormone. Come on! Just one more.
I did it for 5 years, 6 years.
I have a friend with a growth hormone deficiency. I mean, she had a growth hormone deficiency. Now she's fine. She actually had to take supplemental growth hormone as a kid, so she's like 5'7". She's quite tall for a woman.
And my original height prediction was 5'2".
Crazy. Look at that.
Oh, yeah.
My mom is 5'3", but she has scoliosis and she's 5'3", so she was supposed to be 5'6". Okay, so that is our overview of endocrinology. By the way, if you didn't know, FLAT in flat, these are the tropic hormones, and PEG, those are the direct hormones. FLAT are your tropic hormones, and PEG are your direct hormones. What time is it, by the way? 7:50? 7:30. Okay. About to freak out if it was 7:50. Holy shit. Time flies. When you're having fun. Yay! I'm having so much fun. My back doesn't hurt at all. By the way, if you are planning to take the MCAT in April, May, or June, which is most of you, I'm assuming, I would suggest taking a half or full-length diagnostic at some time within the next 2-3 weeks and coming back to me with your score to talk about it. Have fun, and if you're at home, email me or text me. I sign off on my emails with my phone number, which I probably should stop doing because there's like 70 people on that email thread now. But no one's killed me yet. Please don't kill me.
Contradicting what you said earlier.
Good catch. Real quick, what's the difference that and this?
Self-death. Self-destruction.
Yeah, that one go. Yeah, you were trying to do some apoptosis.
Which one's controlled? Apoptosis is controlled, and the MCAT loves the word programmed. Programmed cell death. Programmed by who? The mitochondria. The mitochondria is really, really heavily implicated. Is really heavily implicated inside of apoptosis. How are you going to remember that? Because if you fuck up, your mom wants to kill you. Hello, Laura. You walked in at a great time. It's good to see you again. The whole crew's back. We were talking earlier about how mitochondria come from your mother, right? So mitochondria controls apoptosis. If you mess up, your mom's going to kill you. Necrosis is uncontrolled cell death. And that's where cells just die. Lack of oxygen, too many toxic factors, they just die. There's like a whole process of the cell losing its nucleus and this and that and blah, 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 and it doesn't explode or anything, and it just shrivels up and it dies. This splatter everywhere. There's red sirens and alarms going off and everything. And that's why necrosis usually comes in a group. There's a group of cells that are just fucked because there's so much stuff that just gets tossed out of the cell. Yes.
Do you think we need to know the mechanism?
Like the back's back, like BCL12. No, you don't. No. You will in medical school, but you don't know. A child comes to your clinic with bow legs. Bow legs. Their growth has been abnormal and their height is stunted. Or their growth has been stunted, their height is abnormal. Which of the following is most likely to be high in this patient? First, you have to diagnose the kid. And it's a diagnosis we've spoken about before.
Is it a fat kid?
No, it's a baby. A kid, like a kid. What does the kid have? Hello, everyone. Hello, welcome. Oh, really? I'm Yusuf. How are you guys? As time goes on, honestly, the lectures might get shorter. So we might start at 7 instead of 6. Would you guys mind terribly going from 7 to 9:30 instead of 6 to like 8:30? All right. So we'll push up by an hour. We'll do 7 to 9:30 starting next week. Because I will not be here on Thursday. I have 2 4-hour exams on Friday. No, sorry. I have 2 2.5-hour exams on Friday. I have a 4-hour exam next week. Okay. Huh? Yeah, so these 2 weeks, I have 6 exams over 2 weeks. I had 1 yesterday. I have 1 today. I have 2 on Friday. Well, technically it's more than 6 exams. I have 1 yesterday, 1 today, 2 on Friday, 2 on Monday, 1 on Thursday, 1 on Friday. and then I leave. Then I'm away for a week.
That's parathyroid, right?
Parathyroid hormone. What does the kid have? Yeah. No. You would think it's growth hormone because their growth is stunted, right? But the bow legs, that's a really strange finding. Rickets. The kid has rickets. Good job. What is rickets? The bones are brittle. Why? The kid's not getting enough of a specific vitamin. Vitamin D. Rickets is a vitamin D deficiency in children specifically. In adults, it's called something different. Does anyone know what it's called in adults? Osteomalacia. Osteomalacia.
Which one's the one about osteocytes eat away at your thing?
We will talk about that in a second. What's the answer to the question? Whoa. Hold on. Hold on. All right. Who thinks it's A? No one. Who thinks it's B? No one. Who thinks it's C? 1, 2, 3. Anyone? 4, 5? 5. Oops. Who thinks it's D? 1, 2, 3, 4, 5. 6. Why is it D? Why is the answer D? Well, vitamin D is essential for what? Calcium uptake from small intestine. So if you lack vitamin D, you have low calcium from the intestine, which means you have low calcium in the blood. Do you want to tone down serum calcium when you have low calcium? No. You want to start wasting those bones. So when you waste the bones away and the child tries to move around or stand or move its legs or do anything and the bones are brittle, they flex. And they flex inward and you get bow legs.
That's why I asked you, because I know like obese kids,
Right.
sound like,
No, not at all,
they're
please.
like too fat. Their legs
Totally.
are big.
I mean, that's the #1 risk factor for osteoporosis in the future, obese weight. The #1 reason that people have osteoporosis is because of obesity. Because you're putting more and more and more pressure on those joints and those joint spaces begin to close. Every single time you climb, I think it's either up or down the stairs, you put 3 to 4 times your body weight on one leg, on one hip and knee. That's a lot of weight. That is a lot of weight to put on one joint. I'm 165 pounds. What's 4 times 165? A lot.
So would you recommend just taking the elevator?
No. You guys like this?
Yeah.
Good. Cool. That brings me to another point. Some vitamins that you should maybe be aware of. Not even vitamins you should be aware of, just small facts about specific vitamins. #1, vitamin A. What's it good for? Lots of things. Anyone who likes skincare should know that vitamin A is great for getting rid of pigmentation, right? Retinol and retinoid and retinol derivatives. I just made a video about it on my TikTok the other day, right? But vitamin A, specifically for the MCAT, is really good to make retinal. And retinal is a visual pigment. Vision. Or retinoic acid. I think it's like 11 cis and then all trans. There's like that change that happens when the light irradiates that double bond. And then there's rhodopsin, and the retinoic acid is connected to rhodopsin, and the conformational change of the retinoic acid causes the rhodopsin to change, which activates the G protein coupled receptor. G protein coupled receptor sends a signal through the retina, onto the optic nerve, through the brain, to the occipital lobe, and you see an image.
Is that how retinol works?
That's a retinoic acid. Vitamin B. Which one? Which fucking one? All right, so let's do some really, really quick facts. Vitamin B2 is a component of what? FAD, and FMN. This is riboflavin, right? B2. B3 is a component of niacin, which is part of NAD and NADP+.
Is that why you flush when you take a lot of niacin? You get
Yes, you flush. It's a side effect of niacin. Niacin is also good for LDL control, cholesterol control. B5, coenzyme A. Pantothenic acid. It's part of coenzyme A. Coenzyme A is a huge molecule. Massive. Absolutely massive. B7,
What about 6?
fuck B6. You don't really need to know much about it. That's the thing.
Pantothenic acid, right?
Pantothenic acid is B5. Pyroxidine is B6. What's B7 good for? Biotin is good for hair. B9 and B12, everything. Literally everything, especially having children. I fucked up last time in the video. I like the time that I was talking about B9, B12. I want to correct myself because I correct myself in the sheet. I don't know if you guys look at that all the time. I said that B12 deficiencies will cause neural tube defects, and then I said because the baby eats up the folate. First of all, B12 isn't folate. B9 is folate, and folate is the correct term. B9 deficiencies will cause central nervous system problems inside of the child when they're born. That's why it's kind of recommended that any sexually active woman of childbearing age, hopefully you're of childbearing age if you're sexually active, right, should be taking folate supplements. Because if they get pregnant, you don't want any risk that the baby comes out fucked up. Okay, that's vitamins. That's more biochem than it is bio. Oh, sorry, but that's not all the vitamins. Vitamin D, we said, is for calcium uptake. And where do you get vitamin D from, guys? Touch grass. Go out in the sun, touch some grass. Don't sit at your computer. All my computer science friends are just like, oh, bro, I have to take like 25,000 pills of vitamin D every day, or else I'm gonna shrivel up like a worm and die. I was like, bro, you go work outside. Can you go somewhere? Do something with your life. And vitamin K, which I want to mention to you. What's it good for?
I was going to touch on the vitamin D because
Yeah, what's up?
the professor said that it's a hormone. It's not really a vitamin.
No.
Like there's a specific, is with like deoxy 125 calcium.
Well, yeah, like calcitriol is vitamin D, but it's not really a hormone. Like it's a signaling molecule, like it can be used that way, but because not even. Like vitamins are cofactors. That's the thing about vitamins, that they're cofactors to enzymes and processes. So, vitamin K. What does vitamin K do? Clotting. I think it's if my memory doesn't fail me, 2, 7, 9, 12, protein C, and protein S. And these are all the clotting factors that are dependent on vitamin K. No. I'm flexing on you for no reason. And specifically, warfarin is a drug given that inhibits the regeneration of vitamin K. So it decreases those clotting factors, and that's how it controls coagulation. Who here knows their blood type? Tell me your blood type.
O minus,
O negative. Tell me your blood type.
B plus.
B positive, just like me. AB positive. Anyone else? O positive, just like my dad. Anyone else? They don't tell you? What do you mean they don't tell you?
I've done so many blood.
What the fuck do you mean they don't tell you? Just ask. You ever donated blood before? They tell you when you donate blood. They give you a card. Hold on. I do.
1-800-595.
Honestly, great business model. A lot of people will buy into that. Where the hell? There we go. Blood donor. Be positive. Unless they stop giving those out because they're cheap. Cheap. Okay. So how does this work? What is the inheritance of A, B, and O antigen? Well, first of all, what are they? Right? They are antigens that exist on the surface of your red blood cells. And there are so many of them. There's tens of them, right? But you only need to know a couple. So let's say you have a red blood cell. Now let's say you have the A antigen. And how am I going to resemble the A antigen? With an A. And you have another red blood cell. And you got a B. And then you got another red blood cell. And you got an O. Something's wrong here. Who can tell me what's wrong? Who's been paying attention? The O antigen doesn't exist. The O means 0. 0 antigens. We'll get there. People with the A antigen make what antibodies? Their antibodies are not B, they're anti-B. And hold on, let me draw this a little better so you know it's an antibody and not a Y. Is that good? Okay. What about these guys? Their antibodies are anti-A. And these guys make anti-A, anti-B. All right. How do you inherit them? Well, here's my dad. And here's my mom. What does I mean? O-type blood. And there's my sister. And there's me. That's how you inherit them. So I have a recessive antigen for O-type blood. That's how you inherit them. What about that Rh factor bullshit? What is that? Right. So the only thing you need to know about the Rh factor, right, is that some people are Rh positive and some people are Rh negative. And Rh negative people don't really like Rh positive blood. So much so that if a mother who's Rh negative gives birth to an Rh positive child and their blood mixes, the mother will start creating antibodies against Rh factor. Now the Rh negative mother has antibodies against the Rh factor. So if she gets pregnant again, and that baby is Rh positive, that baby gonna die. Unless you have modern medicine. Yeah, I forget. I totally forget what it's called, but some sort of crazy, very scary disease name. Yep.
So what's the difference between O and then AB, and then what's the plus and minus to?
The plus minus is the Rh factor. So I'm B positive, which means I have Rh factor. And if you're, let's say, something negative, let's say we become blood brothers. I slit my hand, you slit yours, we shake hands, right? Your body will immediately start creating antibodies against that Rh factor in my body. Right. So now let's talk about AB, B, O, whatever. I'm B positive, right? I, on my way home, get stabbed by a midget, and I'm bleeding out on the ground. Because look at how many people are laughing now. So a little 3'6" person comes up behind me, jumps up, and stabs me in my back, right? All right. And now I'm bleeding out on the floor. EMT comes. They're just like, oh my God, he's dying. And the only blood that they have is A-type blood. and they pump me full of it. What's going to happen? Well, I'm B. I have anti-A antibodies. Those antibodies are going to surround the red blood cells. And when the antibodies surround the red blood cells, that's called opsonization, when antibodies flood and stop something. And then my immune system is going to destroy those red blood cells. I can't even use them. I can't use them. There's no point. What if they give me B-type blood? Do I have anti-B antibodies? No, or else I'd destroy my own blood, right? What if they gave me O-type blood? Well, there's no antigens there, so there's nothing to destroy. So I can accept B-type blood, O-type blood, and I'm Rh positive. So that means I can accept Rh positive blood and I can accept Rh negative blood, given that the Rh negative donor doesn't have Rh antibodies or they've been sifted out of their blood, which probably happens when you donate blood, if you do have them in the first place. Okay, what about this guy? Anyone here have A-type blood? No, let's talk about Laura. So Laura is going to walk home and get stabbed by a midget. And she's just bleeding out on the floor. And I'm like, oh my God, Laura, I'll save you. So I hook a needle into my arm and put one right in her left fucking atrium. And I'm just like pushing really hard. And the blood just starts flowing out of my arm and into her left atrium. Is she going to be okay? Why? Because look, A-type people make anti-B, B-type people make anti-A, AB people make nothing. She's going to be completely fine, no matter who gives her blood, especially since she's positive. She's Rh positive. So Rh positive people, Rh negative people, and O-type people, and A-type people, and B-type people, and stupid people, and everyone. Everyone can give her blood. Anyone, right? As long as you don't have AIDS. Don't give Laura blood if you have AIDS. What about O-type blood? You and Sarah. You two. You are both walking home, and you get shot by a midget. Right in the fucking aorta, right? Luckily, a cardiothoracic surgeon just happens to be walking next to you, repairs the aorta, and just grabs a bag of blood out of his car, starts transfusing. Unlucky for you, I'm sitting in the car pushing blood out of my body, right? Unlucky for you. Why? Because you make anti-A and anti-B antibodies. So when I give you my B-type blood, it's gone in seconds. And when Laura gives you her blood with A and B, it's gone in even less seconds. And then when someone else with A-type blood gives you their A-type blood, it's gone in even less seconds, in whatever, right? It's gone, is the point. You can only receive blood from people with O-type blood. Because why? Because you make antibodies against all other types of blood except O. So, the thing is, people with the Rh factor, can we donate to people with Rh negative blood? We could if it's their first exposure, probably, because it takes time to mount that response, right? Maybe they'd be okay, but better not to. So Rh positive people can donate to Rh positive people. Rh negative people can donate to everyone, which is why David is the most valuable person in the room right now.
I'm not saving you.
Man, fuck that guy. David's gonna watch me get stabbed and just be like, haha. I'd probably be laughing too, not gonna lie. Oh, finally! Yes!
And the midget's gonna run away.
Can't run that fast. I mean, come on.
I have strength knowing that.
We did APO antigens. It is. What do you mean? What do you mean? Where did they come from? Do they like crawl out of little holes in the ground? Like what the fuck? It's called achondroplasia.
Midget's watching the video.
Shout out to any midgets back home, bro. We fuck with you. Achondroplasia. Achondroplasia. Who's that Game of Thrones actor? Yeah. I've never fucking watched Game of Thrones. It's just porn. For like for the first 2 seasons, it's just porn on TV.
That's like for 2 minutes each episode.
Nah, bro, astaghfirullah, bro. Come on. Don't do that.
White Lotus is porn. Game of Thrones.
I don't watch White Lotus either. Do I look like I... Do I look like I have time to watch fucking Game of Thrones? I don't have time. Dude, I spend my free time teaching you about midgets, okay? I'm going to go home and study. I have work to do. I have an exam in 2 days. Let's talk about action potentials. Let's talk about action potentials and action potential propagation, because I feel like this is a simple enough concept that too many people forget. Way too many people forget, right? So let's talk about it. Okay. Which way is depolarization? Which way is repolarization? Up is dipole, down is repole. And then below baseline is called hyperpole, right? Okay, so baseline is negative 70 millivolts. Threshold is negative 50 millivolts. Max is positive 35 millivolts. And hyperpole, I think, is like negative 85, negative 80, something like that. Let's just call it negative 80. So at the head of the axon, at the head of the neuron, right, we have a neuron cell body. We have a long axon. And we have all these dendrites. I'm going to pimp you guys about a lot of information about this axon, so be prepared. This is the cell body. This is the axon. These are the dendrites. And the dendrites are part of the presynaptic neuron. There is a neuron following this. If there's a neuron following this, it is called the postsynaptic neuron. If it's not a neuron, it's a muscle, usually. And then it's not called the synapse, it's called the neuromuscular junction. Okay, a neuromuscular junction is a specific type of synapse between a presynaptic neuron and its effector. So, temporal and spatial summation of all bunch of different impulses coming in. If the summation of those impulses is high enough to go to threshold potential, the neuron does what? It opens what type of channels? Sodium channels. Right? Sodium in, syn, right? And then potassium out, pout, syn, pout, right? So we open the sodium channels. Are they fast to close or slow to close? They are fast to close or be inactivated, right? Then as we come here, the neuron needs to reset everything, right? Because we have this massive influx of positive charges, so we're going to push a bunch of positive charges out in order to get back to resting potential. So in an effort to do that, it opens potassium channels, right? And what is it that forces all this sodium in? Is the sodium high outside the cell or inside the cell? Sodium is high outside. So there's a lot of pressure against that sodium channel, right? It's like, you guys ever been out in like Williamsburg or downtown Manhattan on a Saturday night, and you see like, here's the door to the shittiest club you've ever seen in your entire life, and there's a swarm of people. If they open that door, boom, gone, right? Everyone's going in. This is the club. This is the club, and the sodium's trying to get in. And the moment the sodium gets in, the potassium, the smart guy's like, I'm getting the fuck out of here. I'm done. I don't want anything to do with this, right? So when you open the potassium, there's a lot of potassium in here right now. You open the door, boom, it goes the reverse way. But The potassium channels are what? Slow to close or quick to close? Slow to close. So they overshoot the balance. They overshoot that resting potential. And now you got to do work to get back to resting potential. And that work is done through what? Sodium, potassium, ATPase. Sodium potassium pumps. That's it. That's the whole shebang. Right. Now, I'm going to pimp you guys about the neuron. What are these?
Myelin sheath?
No. What are these? No. Nodes of Ranvier.
That's not the Schwab cell things, right?
We'll get there. The nodes of Ranvier. The nodes of Ranvier are the unmyelinated sections of the axon. The unmyelinated sections of the axon. Because, right? What is myelin? It's an insulator, right? It's basically made of fat. Does fat conduct electricity? No. No. Fat does not conduct electricity, right? It's just a bunch of, it's made up of a bunch of lipids, sphingomyelin, right? Sphingolipids. So it's not going to conduct the electricity. But one thing that does happen is that when the electrical impulse begins, it's going to hop these nodes of Ranvier. It's going to hop the nodes of Ranvier. And that's faster. It's not just faster. Here's the interesting thing. They always teach you on the MCAT that it's faster. It is faster. But it's not just faster. It's more efficient. Because what's going to happen, right? Let's say I'm the action potential, right? And let's say from here to there is a mile. From here, this side of the room to that side of the room is a mile. And I have to sprint the mile as quickly as I possibly can, right? So when I begin there, I'm full speed ahead. But what happens when I get here? I'm slow, I'm tired, I'm exhausted. So it's possible that if we were to travel down the entire axon, we would lose some of the energy of the potential. We want to conserve as much of the energy in the signal as possible. So we jump between those nodes. So the actual distance that's traveled is far, far, far less than the actual distance of the axon, number one. Number two, sodium and potassium all along here. What's the important ion there? Calcium. There's calcium release that allows for vesicles filled with neurotransmitters to associate with the end of the neuron and be released into the synapse. This is the cell body, correct? Right here, this general area, what's it called?
Soma?
The soma, yeah, but the axon hillock. The beginning of the axon, where all of that summation takes place, right? One more thing. If this neuron is in the central nervous system, what type of cells are making the myelin? Oligodendrocytes. These are the cells that are destroyed in what disease?
Alzheimer's.
No.
Parkinson's?
No.
MS.
Yes. Multiple sclerosis. Multiple sclerosis is a central nervous system demyelinating disorder.
That's what it's
And it comes and it goes, and it comes and it goes, and it comes and it goes. If this were in the peripheral nervous system, what cells are affected? What cells make the myelin in the peripheral nervous system? Schwann cells. What disease or disease process destroys Schwann cells? No.
SMA?
Guillain-Barré. Guillain-Barré.
What about diabetes?
It can. Diabetic neuropathy, yeah. Diabetes is a lot of things. Nephropathy, retinopathy, neuropathy, diabetic foot disease, blah, 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 blah. Yeah. I'm so bad at that stuff. I'm not going to lie to you. Like, the only thing I know about the refractory period is that it's like the sodium potassium pump action. The absolute refractory period is absolutely no signal can be transmitted. The relative refractory period is like, okay, we've garnered enough stability. So if you kick me in the fucking balls, I'll do something. Like, normally you just need to like poke the neuron to do it. If you're in the relative refractory period, you have to like stab it, and then it'll do something. Don't stab neurons. Do not stab yourself in the head. Great advice from Yusuf. All right. One final thing that we'll go over, and then we'll chat as a group for a couple minutes. And then you guys can go home, I promise. This is not a sperm cell. It's bacteria. The bacteria enters your body, right? And it's foreign. Who's going to eat it? Probably a macrophage. Nom nom. Right? A macrophage, also known as an antigen-presenting cell. It's going to chop up the bacteria, right? Preserve its antigen, and use what to display it? MHC class 2. Why 2? Because 2 is the exogenous pathway, outside. And the MHC2 is recognized by what T cell? CD4, which is the helper T cell, which is going to blow a horn and cause B cells to copy the antigen, go to the lymph nodes, replicate the antigen, attach it to made antibodies, produce antibodies against that bacteria, produce the antibodies, send out the antibodies, opsonize the bacteria, and terminate the infection. Rather, let's say a virus gets in your body, and it takes this is your cell, it takes its DNA and it injects it into your cell, and it begins assembling viral particles. Well, before those viral particles get out, your cell inside has another histocompatibility complex in it. What's it called? MHC? 1. Those viral particles get chopped up. The antigens get attached to MHC1 and are produced and presented on the cell. And that production and the display of the antigen activates what? CD8 T cells, killer T cells. And they're going to blow the horn and be like, holy shit, you need to die. You need to die, right? Why? Because if you don't die, then you're going to die and leak all of this virus all over the body. So the CD8 T cell comes, injects the cell with pro-apoptotic factors, boom, dead. And goes on to recognize more of those antigens, and then boom, dead, boom, dead, boom, dead, boom, dead. And some cells can even, when they have, some viruses are so smart, but we're smarter. Well, maybe, sometimes. Some viruses will downplay MHC1, will decrease the transcription and ability of MHC1 to do its job. Any cell with downregulated MHC1, dead. Any cell with downregulated MHC1, dead, gone, because you can no longer do your job. Your body is the strictest factory in the entire world. You can't do your job, done. Finished. Huh? NK cells, yeah. And that is it for Biology Review. Thank you guys so much for watching. Everyone here, stick around for a few minutes.